Now, we do learn about this polypeptides, a primary structure. So who determine the primary structure? Because when we talk about primary structure, we look at what are the amino acids and what is the sequence of the amino acids. So who determine? Our DNA, our gene is the one that determines, okay, the sequence of the amino acid. And this we're going to learn them in the transcription and translations of the during the protein synthesis or polypeptide synthesis process. Okay, so now, for example, how we read our DNA. So every DNA triplet here, for example, CAC, GTG, GAC is a code. So this code will be used for us to put in the correct amino acid, join them by the peptide bond. So under normal conditions at our beta polypeptide, because we do know that our hemoglobins have 4 polypeptide chains. So hemoglobin again is a quaternary proteins. Okay, so one of the chains is called beta polypeptides, okay, or beta globins. Okay, either way you can use either beta polypeptide or beta globin. So these beta globins at the 6 positions under normal conditions, no mutation, is the glutamic acid. And glutamic acid is a negatively charged R group. They are hydrophilic. Okay, they are hydrophilic. Okay, because CTC instructs, say that, okay, because I'm CTC, so it's glutamic acid. So you have a normal hemoglobin, normal beta globin. Are you clear? At the 6th position, 1, 2, 3, 4, 5, 6. At the 6th position of the beta globins, for normal hemoglobins, it will be glutamic acid. But because of single base substitution, for the thymines become the adenine. Can you see that? T become A. Can you see that? So the code means wrong already. Okay, you try to imagine it's something like the coordinates, and you put in the waist. Okay, the coordinates. So you put the wrong coordinates, so direction will be different. Same goes to here. CTC becomes CAC, then it changed. Okay, the instruction given here will be glutamic acid become valine. So this valine right now is non-polar. Okay, it's a non-polar amino acid that are grouped. So what will happen here? So we do know that hydrophilic R group maintain the solubility, but now we change the ones that can interact with water molecule, the hydrophilics, with the non-polar hydrophobics, valine, therefore decrease the solubility. So what will happen here is, this is normal red blood cell. So normal red blood cell, you can see that our hemoglobin, because they're soluble, they become the molecular form. They scattered inside our red blood cell. Are you clear? So this is normal. But for sickle cell patient, under high partial pressure of O2, let's say, for example, in the lung, because you breathe in the fresh air, so it tends to be okay, no problem. But when they proceed into the muscle cell, when the PO2 partial pressure O2 is low, this partial pressure O2 is high. So let's say, for example, in the muscle or liver, what will happen here is, because when the partial pressure O2 is low, this hemoglobin, they tends to precipitate out and form aggregate. They join together like this. So means that when they become longer and longer, it tends to push the red blood cell. Now red blood cell becomes sickle shape. So this is normal shape. This one is called sickle shape. Can you see that? Why? Because it continue. Can you see that? Are you clear? You try to imagine that you put a rod in the center, it tries to bend. So this causes sickle shape. Okay. Now what will happen? 2 things. First, because of this shape, they will cause the vessel occlusion. Now, what's mean a vessel occlusion? You look at this. Normal shape, they can pass through. You can squeeze through, elastic. But here, can you see that it blocked at a small capillary? So it means that the organ won't receive sufficient oxygen. So this one we call a vessel occlusion. It blocked. And because of the vessel occlusions, the organs or the cell or tissue will face what we call the ischemics. Ischemics basically means that lack of oxygen for them to survive. Are you clear? And because these red blood cells, okay, normally the lifespan of red blood cells will be 90 to 120 days. Okay, but because of this, the continuous episodes of, okay, continuously normal, sickle, normal, sickle, normal, sickle. So this red blood cells membrane will lose the elasticity, no longer elastic. So it means that easily it will break. When it break, when hemolysis take place, means that your body won't have sufficient red blood cell. So therefore, anemia take place. Are you clear? So anemia is a disease. Sickle cells anemia is a disease. Okay, but sickle shape is the shape that we describe when you have this precipitate form. Okay, so this precipitate we call it as a fibrous precipitate. So it tends to form a fibrous protein like this, fibrous, and make the shape of the red blood cell become sickle. Okay, sickle shape. Okay, so 5 minutes. So you can, after you have done, you can compare your answer. So I go through slowly the answer. You can compare your answer here. Okay. So the idea of these fibrous precipitates because low in solubility. Okay. And then what is mean vessel occlusions? Vessel occlusion means that the blockage to the blood capillary, and because of the blockage of blood capillary, therefore the cells lack of oxygen. Okay, so therefore ischemia take place. So but the thing is, at this moment the cells, okay, what we call this, it's not anemia yet until the cells is lysed, or we call hemolysis take place. And because our bone marrow produced red blood cells is not fast enough to replace those damaged red blood cells, therefore anemia take place. Okay, so this diagram shows us the vessel occlusions and how the sickle cells actually look like. Okay, so with this I've done for the proteins overall. Okay,